gonna be fine. Everyone was really excited. Jen came in.
Jen.
They were taking the coffee maker because the coffee maker doesn't work in Jen Biddle. And,
Oh, what Jen Biddle is there?
they had, no, it's, everyone was happy.
Huh. Huh.
Everyone was happy.
You did? Oh, I'm sorry. I didn't have to get you with that. No, it's okay. No, I don't. Okay, stop. Oh, okay.
Yeah, so there's gonna be a whole bunch of things on Tuesday.
The next Tuesday? I'll be here. Yeah.
Really?
Yeah.
I know you have
Class at, what's Tuesday? I have class at 2 on Tuesday. I'm open on morning.
Okay, because we could just go. I told them that we just have to pick up with them here. So it's gonna be a lot of fun. I don't know.
Yeah, I'm free on Tuesday. I think Bill and I we're starting my research project. We went out on Monday, but I should be. You ready?
Sure.
Oh, coffee motion? Let's see. All right. I'll email you. Just let me know what time. Sorry.
Yeah, I need to see it anyway.
You wanted to
I
put
know.
it in one?
Yeah, it's so weird. Bill and I went out with Kyra. Let everything from her.
It was designed on this, so I know theoretically it should work
Yeah.
on
The
this.
entire time it's filtering. It's like, it's not cheating. He's like, oh, we'll just figure it out. And I was taking it apart, and I like called up. And I was like, Dr. Ullman. He's like, what? And I was like, can you take the cap off? He was like, sure. I spent like 3 hours like calibrating it. And I was like, we did all this hard work. It took me a long time to do the samples. No, it's fine. I was like, well, no, next time. But just let me know what time.
The director? Okay.
Okay,
So,
you're all good, right?
Let's do this.
let me know how it's.
Yeah. Where in the world that picture come from?
I don't know.
I don't remember this one.
I took it for.
Oh, I was like, why isn't this working? You'll plug that in.
Hi.
Yeah. Kathy's my stepmother Brenda and my father Earl. Like,
I met you a long time ago.
Yeah, I think, yeah.
It's been a while.
I remember, yeah. Well, you're welcome to sit wherever. We'll just punch wherever won't be in the way. Yeah.
I know that he has some colleagues who wants to lunch.
Yeah, it usually gets pretty full for the students.
there we go.
So,
All right, it works.
Yep, so find a.
What do you mean it works? It better work. What are you talking about?
It's not
Otherwise, I'm just going to talk. You can use your imagination.
Okay.
Use your imagination.
We've been doing that with you for quite a while.
Yeah.
That's what we are going to run. My friend was saying that, let's see. I can see everything. Excellent. Yeah, Katie's in a meeting right now, so she wanted to see it, but she's going to, and it records it and everything like that. So. I got signature pages.
Okay, good. Yeah, I just thought of those today.
And, oh, no, yeah, don't worry. I double checked.
With one of Katie's committee members, Texas, so she had to
Vet-
FedEx
yeah.
that and then get
Yeah.
it back.
Yeah, and so I have like 500 reams of the special paper.
Mhm.
So whenever it has the UD
Oh, yes.
watermark on it and everything like that. So,
I don't know how many copies are required.
3, but I printed out an extra one just in case. And then there's this one. trying to figure out what I'm doing there.
You're pipetting. You're wearing your safety glasses.
Oh, so that means someone must have been in the. They must have been in the lab filming us or
Yeah,
something.
either that or because I got my hand slapped for not wearing a picture once.
See, I got the doll's pictures right next to me.
Yes. Keep calm and redo everything.
Yes, you put on my. Yeah, I followed that pretty well.
Yeah, she did.
I did redo a lot of things.
Yeah.
Uh-oh, we got
Interview and see blog up there.
Oh, you should put. I have one for science as well.
Do you?
Yeah, it's
I
science.
never saw that.
Yeah.
I've only seen this one.
Here's the hat.
Yeah.
That looks familiar.
Right.
I had a few more that you could have slid in there.
I gave her a couple. No. Could have given her one where I photoshopped my face on Mario or Yamir Yonkers. He played the Penguins, had this huge mullet. So I was gonna go for Halloween wear a mullet with my hockey equipment. Yeah, that's pretty bad.
I had lunch with Diane, so she was happy.
Okay. I saw Ty last night. He's like, "Yeah, I haven't seen Mark in a while." I'm like, "Uh-oh." He's in the country, right?
He said, I sent him an email this morning. Good cookies.
Cookies and I'm not sure what anime.
She's not gonna be here, so
She made something up. Left it for She said she'd probably be back for a quarter.
okay. How's it feel?
I'll let you know when.
You know, we didn't think we should have had some like cold beer and chili.
I asked,
Telling me he was
I
gonna
would bring it, and she told me no. She's like, no.
I give the wrong impression. There is,
There is a
right
happy
after
hour.
this, there's happy hour downstairs in the, in 104 in the Colby Room, so. We'll have beer and wine available there.
We were here for another party.
Yes.
Is this your other committee member?
Oh, that's just me.
Yeah.
You can. I'm just
He's,
streaming it to
he's
friends
streaming.
and stuff like that.
Oh, okay. So Diana, is it? Is she in person?
No, she's gonna be here.
That-
But he had some other friends who wanted to see it,
Yeah.
so he's got it set up with a webcam, so
Okay.
it streams the whole thing. But you can see.
But she is in person.
Yes.
Okay.
Do you know how to operate the ITV thing?
Mhm.
Can you do that for me? 'Cause I'll mess it up.
Do you want to be connected to 206?
Yeah, I don't know if anybody's gonna be up there or not, but let's go ahead and connect and then if
Is
nobody's
that the
there, we can-
206 would be the.
I think so.
There's meetings in the other rooms.
Up there, there's a meeting? Oh, okay, well, maybe we should just not connect there.
No, in the other rooms.
Oh,
Oh, I'm sorry, okay.
Sorry.
They
Draw a
probably-
crowd.
Yeah. Oh, yeah.
If no one comes, no.
Yeah.
I'll give it like
nobody-
5 minutes.
Yeah, nobody shows up.
And of course, someone will obviously come up in the middle of the. What I like to call in the middle.
How was the
of
faculty meeting, Gary?
How's the faculty meeting?
This close to quitting.
That good, huh?
In general, of course, we got nothing done. There was some lovely things inside. Yeah, we should go talk about it.
Last time the meeting was at 2.
Yeah. Hello.
How are you? Just bring their computers. It's fun.
Oh, no, that one's for me because
Yeah,
I'm streaming out to some
He's
friends.
streaming. Awesome. He's got it on the internet.
I've been told Rachel and Andre joins.
Yeah.
Andre's on mute. Better than me.
Oh, no, they can't say anything.
We can sit wherever. And what we usually do is after his presentation, we'll have a short break and then we'll come back in. And then if you want to move up or sit where you want, wherever you can do that.
Throw the rotten fruit. How's the Hotel Rodney? Is that the one right next to Rose and Crown?
Yes,
Yeah.
it is.
I've never been in there. How is it?
It's actually really nice. The rooms were a little on the, a little on the small side.
Yeah.
But, it's nice. Rooms are warm. Get hot water. The heat works. The internet's good. It's
Right.
kind of what I was
Things.
after. So
Yes.
it's nice. It kind of has this sort of like, African safari theme about it.
Really?
Yeah, it's a little strange. But,
Yeah.
I don't know. It's cool.
I always thought it was associated with the Rose and Crowns. I always thought it would be more of a kind of a British,
Like a British
UK,
pub?
England theme kind of. But yeah.
I don't know.
Sorry, it came up with the cold weather though. It's supposed to be a little warmer. Next couple
Yeah.
days.
Actually, the flight up was a little bumpy because of the air mass coming down. It wasn't as bad as I thought it might be, but yeah, I mean, it was freezing. It's actually almost the same temperature in Charleston. I mean, it's freezing there too. I'm gonna just hop in the restroom. Is there a restroom?
Yeah, if you go down this hallway and then it's the first left, there's a sign. Kind of
Rachel says hi. Apparently she can hear us just fine.
All right, excellent. And can she see the video? Because my friend said she couldn't see the video.
Tell us if you can see the video. I know you can hear me.
Oh, yeah, I guess.
Find out.
You should have her show Kevin. She's working with Kevin for the next month. Selling her?
Oh, okay, yeah.
So, yeah, he's only gonna be there to the end of the year. So. What did you say you were? I'm sure I'd be a hard sell for him. Anybody.
I'm on the committee.
Does that mean I have to bring that slide for everybody on the committee or just you?
Okay, well, that's what I'm saying.
And me?
I think Kathy needs something strong.
Rachel says she can see us.
He said he was gonna put a bucket of beer outside.
You vetoed that. I would have.
I vetoed it,
And then
I
I told him
would've-
how
there
other
would've
schools are
plenty of
actually
beer out
disallowing food so that no one is swayed anyway.
Just run down and empty out during that 15-minute break.
Well, that's what Doug was saying. It's like, there's a fridge full, it's just gonna get it.
Wait for the show. Anybody seen Mark?
No. No.
I thought it started at 3, so I was super early. I was giving Marsha crap when she had made the coffee at like 2:58. I'm like, doesn't it start at 3? Why isn't the coffee made?
Chris, I'm sorry for this.
What's that?
Andre says you can hear me.
He's good. He shouldn't be.
Apparently, I'm your connection point here.
Got it. Yeah, 'cause my phone's buried. Sorry. Tom, you see Mark? Okay, you won't be long. All right.
And you have a package in the loading dock.
Someone's up there.
How you doing? How'd you get our lab slogan in your photo?
What's that?
You got our lab slogan in your photo.
It was,
Keep calm and redo everything.
Kathy printed out at the same time I think you did.
Keep calm.
calm and do everything.
Okay, guys.
It just seemed so appropriate that year. It's now, I think, our official motto. Apparently, we're live feed
Who's on the air?
people.
Chris Main Tank.
Chris Main's fan club is on the other.
Yeah. I was gonna post it on Twitter, but I decided against it.
I went old school.
It's okay. Just the forest to
I
cry.
already went through a pair of contacts trying to read them. I was like, two pages. Yeah.
Yeah, it's just
It
like.
doesn't mean
it's
they
just
can
like-
carry it with me anywhere. It's just going.
No magical thing gets worse.
All right, I'd like to welcome you all to Chris Main's PhD defense. Chris got his BS in marine biology from the University of Western Florida, and then his master's in biology from the same institution. Chris started here in 2010 for his PhD. Before he even came for his interview, though, I ran across his blog page, Beer and Seaweed. So I figured that he would probably fit in very well.
I'm glad it's on.
Chris has had an opportunity to present his research at a number of national and international conferences, where he was able to also sample the local brew. And as for outreach, Chris never passed up an opportunity to share his love of science and his passion for algae, sometimes going above and beyond, as you can see here, by wearing his red tide harmful algal bloom cap when he participated in the polar plunge a few years ago. Now, Chris has also been very productive as a student in the lab. He's already published 2 papers on part of his research, comparing quantitative real-time PCR to another molecular method, the sandwich hybridization assay. He has one paper published as the first author and another paper published as the second author. Today he's going to be talking about a different aspect of his research where he is comparing, where he is evaluating the interactions between pathogenic bacteria and harmful algal bloom species in Delaware's inland bays. Before he starts, though, I do want to introduce his committee members. We have Jen Biddle and Mark Warner from the University of Delaware. Diane Greenfield is Chris's outside committee member, and she is from the University of South Carolina. All right, and with that, I will let you take it away.
right, so can everybody hear me? Make sure I'm projecting well enough and things like that. Well, thank you for coming to my thesis or dissertation defense. As Kathy mentioned, today I'm going to be talking about the molecular assessment of rhipidophyte species and their associations with Vibrio in the Delaware Inland Bay. To begin with, I'm going to give a little bit of background information about Vibrio. So Vibrio are marine bacteria that are ubiquitous in the marine environment, and there are several pathogenic species to humans. The three major ones people are probably very familiar with is Vibrio cholerae. This is the causative agent of cholera. Right now, we've been in a pandemic of cholera since about 1960, and the country of Haiti has been dealing with issues of cholera since about 2008 and their earthquake there. There's also Vibrio parahemolyticus, which is the already messing up already. Which is the leading cause of seafood-borne illnesses worldwide. And in addition to that, there's also Vibrio vulnificus, which it causes about 95% of the seafood-borne illnesses here in the United States. So just between these three particular species, these are a very important aspect that need to be studied in the marine environment. However, you shouldn't worry too much about it. While they're ubiquitous in the marine environment, the percentage that is pathogenic of Vibrios is very small. And the number of Vibrio species in the entire bacterial community is also very small as well. But they still make up a very important members of the marine environment. Now, as I mentioned, these are very important species that we need for human health issues. And so since about 1996, they've been, this is looking at laboratory-confirmed infections by Vibrio. And we've been seeing a nice increase of Vibrio cases in the United States since about 2000. And in fact, in the last 3 or 4 years, we've actually doubled the number of Vibrio infections here in the United States itself. So this is very important that we understand the ecology of these organisms in the environment. Now, if we narrow it down here to the Mid-Atlantic region, we actually see that the Mid-Atlantic region has a high incidence in the number of Vibrio cases, as presented here on this map. But illness is just not by ingestion from contaminated seafood. You can actually get it just from a simple scratch swimming in the environment and things like that. So what is the main driver of these increases in Vibrio cases here throughout the world? Well, the major driver is actually going to be sea surface temperature. So as sea surface temperatures, indicated here by the yellow line, increases, the number of reported Vibrio cases also increases. So as we begin to have global climate change occurring, and with the sea surface water increasing, we then begin to see increase in the number of Vibrio species, or Vibrio diseases, sorry. Now, Vibrio and other marine bacteria exhibit two major growth stages or growth strategies within the marine environment. They are free-living members of the bacterial plankton here highlighted in red, or they may associate with particles as a biofilm, and we are beginning to see the beginning stages of biofilm formation there on the bottom image. Now, there's also this association that has been noted in the previous research that there is an increase in total number of Vibrio that has been linked to phytoplankton biomass. However, there's little known between these interactions, between the species-specific interactions between Vibrio and phytoplankton. Now, if we look at phytoplankton within the marine environment, there is this wonderful term that was established in about the 1970s called the phycosphere. And this is a boundary layer that surrounds the algal cells that is nutrient-rich, that we actually see attachment of bacteria or bacteria associated with these algal cells. So you can see here this nice plume of kind of pinkish orange surrounding the diatom cell, and an artist's rendition of the bacteria swimming towards it. So this nutrient-rich environment allows for proliferation to occur, as well as it may also provide a refuge from grazing from microzooplankton grazers. Now, the main aspect of phytoplankton that I'll be talking about today is a group of algae called the rhipidophytes. These are globally distributed, unicellular, that tolerate a wide range of salinities. Now, they have been linked to fish kills, so they have been determined to be ichthyotoxic. However, the potential cause of their toxicity is kind of up in the air. Some studies have shown that reactive oxygen species may be the culprit for this, as well as polyunsaturated fatty acids, hemolytic compounds, and even a Breville-like compound that is being produced. Now, there has also been other previous research where they have examined the interactions between Vibrio and rhapidophytes, however, this was in laboratory conditions. And they noted that reactive oxygen species produced by rhapidophytes inhibited the growth of Vibrio as well, but interesting enough, exudates from Heterosigma, one of the rhapidophytes, actually caused a positive chemotactic response in a Vibrio algiolenicus. Now here in Delaware Inland Bays, there are 4 species of rhapidophytes that have annual blooms here. They come in roughly 2 size classes, those greater than 20 microns in size, Chatonella subsalsa and Fibrocapsida japonica, and 2 other species that range between 5 and 10 microns in size, Heterosigma akashiwo, there on the image there on the left, and Viribulobus marinus. Now, as mentioned in my title and just in the last slide, my focus of study here has been Delaware's inland bays. And Delaware inland bays consist of 3 interconnected bays with a single outlet. We have the Rehoboth Bay, Indian River Bay, and Little Assawood Bay, and a single outlet here to the Atlantic Ocean that is the Indian River Inlet. And as you can see here, illustrated by this map, we see a lot of developed and farming areas as part of this watershed. And because of that, we see a lot of eutrophication and excess nutrients coming into the environment here. Since about 1991, the Citizens Monitoring Program has been actively monitoring the inland bays. They've been collecting weekly samples, and because of them, and they are a very important aspect of my project here, they've brought in samples from 3 sites throughout the inland bays. RB64, which is part of the Rehoboth Bay, IR32, part of Indian River Bay, and SB10 East, which is part of the Little Assawood Bay. Now, these 3 sites here are what we call dead-end canals, as you can see here, image of the inset, and this is a common feature that we see throughout the inland bays. And these dead-end canals allow for very low flow of water, so they're very stagnant for most of the year, and we see very large, dense blooms of both dinoflagellates and rhapidophytes. Here is an example here of a very dense bloom. Unfortunately, it doesn't really show up too well, it looks like there. But this is a bloom of Heterosigma that I collected, that's my hand right there, of approximately 10 to the 9 cells per liter. And if you look closely, you can actually see some of the streaking there. And one of the aspects is, if you also notice, it's kind of a mucousy-like attachment there to my hand. So if we see this, looking at with Vibrios and things like that, and we see these very strong blooms, it is very important to understand these interactions here. So what are my objectives for my research here? So the first objective was to examine this relationship abundance between algal groups and their associated Vibrio species. In order to do that, I had 3 questions to answer this objective. Is there a correlation between partically associated Vibrio and algal abundance in Delaware's inland bays? Is this association between Vibrio and algae species specific? And finally, does this association provide a refuge from grazing for microzooplankton? The second objective that I wanted to look at with my research is to look at this, investigate the species-specific interactions between Heterosigma and Vibrio. And to answer that, I had these 2 questions: which species of Vibrio were associated with Heterosigma? And finally, what effect does association with Vibrio have on Heterosigma itself? So to answer this first question, this correlation between partically associated Vibrio and algal abundance in the inland bays, the Citizen Monitoring Program brought in weekly samples to me from May to September, from 2009 to 2007. Pardon me. This water was size fractionated and collected onto a 3-micron filter to look at the partically associated Vibrio species. Then I looked at relative abundance of both the algae and Vibrio using qPCR, and the qPCR was looking at total dinoflagellates, so all dinoflagellate species, all diatom species, all rhipidophyte species, and all Vibrio species as well. So since as I said that there has been a strong linkage between sea surface temperature and Vibrio abundance, I also looked at the abundance of partically associated Vibrio, which is on the y-axis. So the next few slides we'll be looking at roughly the same layout here. So on the y-axis is the relative abundance of partically associated Vibrio. And then we have temperature, salinity, and the log transformation of chlorophyll. Now, as I mentioned, with the sea surface temperature, we do not see a strong, or I don't know what, sorry, it's supposed to be boxes coming up. All right, anyway, we do not see a significant correlation between temperature and a partically associated abundance of Vibrio. However, we do see a very significant, or a significant correlation between salinity and the chlorophyll or total phytoplankton community. Now, if we look at this association between, the correlations between partically associated Vibrio, once again on the y-axis, and on the x-axis we have dinoflagellates, rhipidophytes, and diatoms, we see that there's a significant correlation between all three algal groups, but we see stronger correlations between the rhipidophytes and diatoms. So using that information, the next step was to go in and look at the species-specific interactions. So in order to do that, I did intensive sampling with replicate sites over 5 time points during a mixed rhipidophyte bloom. And this bloom was between fibrocapsa and heterosigma. Once again, this was size fractionated. As I mentioned earlier, fibrocapsa is a little bit bigger than 20 microns. So 20 micron fraction will be fibrocapsa associated Vibrio. The 3 micron to 20 micron will be our heterosigma associated Vibrio, and then 0.2 to 3 is going to be the free living ones. Once again, relative abundance was examined using qPCR, and this time I used species-specific probes, primers and probes for heterosigma and fibrocapsa to examine their abundance, but once again used the same general all species encompassing primers for Vibrio. So if we look at the changes here on the graph, on the y-axis on the left is the change in concentration of heterosigma at the start to the end of the bloom, and on the right is the fibrocapsa concentration. You'll notice that there is a 100-fold difference in amount of cells per liter, and we can see just here is just showing you what is occurring with the number of cells during this intensive sampling that I did. Now you also see that fibrocapsa did decrease and actually was not detected after 70 hours, or roughly 3 days of sampling. So if we look at the correlation between partically associated Vibrio and the rhipidophyte species, we see that there is a very strong, significant correlation between partically associated Vibrio and heterosigma. However, there is a negative correlation that is not significant between the Vibrio and fibrocapsa. So the question is, if we're seeing this intensive sampling, is what is happening to the community during this time period? So in order to do that, I analyzed the community, Vibrio community, using a technique called automated ribosomal intergenic spacer analysis, or ARISA. Very briefly, what this allows you to do is you're looking at the intergenic spacer region between the 16S and 23S between these organisms. And each organism, the idea is that each organism, that intergenic spacer region is different in size. And when you use primers that have been modified with a fluorescent probe, you can run them out on an instrument and look at changes in the abundance based on peak height and difference based on the migration of the base pair fragments. So using ARISA to examine the intensive sampling here, this graph here is showing a technique called multidimensional scaling. And this is just a way of visualizing the similarity between samples. Each point that you see on the graph here represents the entire community for that particular sample. And then the closer the points are to each other, the more similar that they are. Now, this should be noted that this is not a not a statistical analysis. This is more just a visual representation to give you an idea of what is occurring in these samples here. But what you should notice is that there is a clustering of the free living, which is the circles here on the left, on your right, and the triangles is the 3 to 20 micron of the heterosigma. So this here is demonstrating that there's a very close similarity between these community structures over time. Now, of course, to examine this similarity, there's another multivariate technique that you can use called analysis of similarity, or ANOSIM. And this allows you to test the various null hypotheses. So I looked at 3 null hypotheses. And my null hypotheses is that there was no significant difference between size fractions, time, or replicates. Now, using this ANOSIM, I did see that there is a significant difference between size fractions, but there was no significant difference with time or replicates. So that tells us is that there is a significant difference in the Vibrio community based on size fraction, but not over time, and not between the 2 replicates that I took. So the next question is what is happening? Why are these Vibrio associating with these algal species? So the next experiment that I did was looking at grazing. Does grazing provide a refuge from microzooplankton? Once again, samples were collected during a different mixed rapidophyte bloom. As it happened, it just happened to be another heterosigma fiber capsule bloom. Here I diluted. I took site water, filtered it through a 0.2 micron to remove any kind of bacteria and everything from the water. And then I diluted the site water, the unfiltered site water, with that to 25%, 50%, and 100% of whole water. And the idea behind this is, illustrated there on the right, is that as you separate the predator and prey from each other, the grazing rate should be lower as they encounter each other less frequently. Once again, these samples were size fractionated, and growth was determined using qPCR. And I actually did this twice during the same bloom. I should also say that these were incubated for 24 hours before the samples were taken. So for the first grazing experiment, here we're looking at, on the y-axis is the apparent growth rate per day, and on the x-axis is our dilution factor. On the left is the 20 micron, 3 and the 3 to 20 in the middle, and then of course the free living. So one of the things that we want to look at here is the grazing and growth rate. So what you should notice here on the 20 micron fraction is that those Vibrio associated with those particles in the greater than 20 micron fraction have a higher growth rate and grazing rate. So this is telling us is that they are attaching and actually have a slight advantage because they're growing a little bit faster than they're actually being eaten or being grazed upon. However, if we look at the 3 micron to 20 and the free living, we see that the grazing rates are actually much higher, and in some cases, such as the 3 micron to 20, almost double the growth rate. So we are seeing here with this first grazing experiment, there is actually an advantage for attaching 2 particles to avoid that grazing. If we look at the second grazing experiment taken during the same bloom, we see things are slightly a little bit different. We see at the 20 micron fraction that the grazing and growth rates are actually very similar. But once again, we see the growth is higher than grazing. In the 3 micron, we actually see the grazing and the growth almost tripling in the amount for both of them. However, you'll notice that in the free living, that the growth rate is almost double the grazing rate. So this is suggesting at this particular time point, it actually might be better to be a free living bacterial plankton. So once again, the question is, what is happening to the Vibrio community during these grazing experiments? So once again, I analyzed the Vibrio community using ERISA. And just to sum this one up a little bit, I'm going to talk about what I call the winners and losers. So with the ERISA, we're looking at the OTUs, it's our species here. These are the peaks that appeared on the instrument. So what you want to really focus in on is that there are some species that are actually benefiting from attaching, and some species that are actually losing from attaching. To illustrate this point, we can take a look at this image here. So this OTU here at 318 and the 20 micron fraction after 24 hours decreased. However, in the 3 to 20 micron fraction after 24 hours, the abundance actually increased. So it's by switching or by associating with that 3 to 20 micron fraction is actually benefiting itself, benefiting survival. Whereas if we look at this OTU at 471, we see the opposite is occurring. We see that after 24 hours in the 20 micron fraction, the abundance is increasing, whereas in the 3 to 20, it is actually decreasing after 24 hours. So to conclude this first part here, is that with the abundance of partically associated Vibrio, there's a strong correlation between to the abundance of rhaphodophytes and diatoms. And during that intensive sampling, if we break down that rhaphodophyte strong correlation to the rhaphodophytes, during this intensive sampling, there's a strong correlation to heterosigma abundance, but not to fibrocapsa. And finally, we actually see with the grazing experiment that there is a species-specific associations between Vibrio and algae, and that can actually provide some refuge for some Vibrio species. So the implications of this is that harmful algal blooms of heterosigma may actually provide a vector for Vibrio pathogens. So the next part of my research was to investigate this species-specific interactions between heterosigma and Vibrio. So based on all this previous data, I'm now looking at what is occurring with heterosigma and Vibrio. So in order to do this, I did 2 different experiments to look at what species of Vibrio were associating with heterosigma. And for this first experiment, I collected during a bloom of heterosigma, isolated heterosigma using flow cytometry, and analyzed the Vibrio that was attached to that isolated heterosigma. In order to do that, I used a technique called temporal temperature gradient electrophoresis. And this is just a community molecular fingerprinting technique where each vertical lane represents a sample. In this case, the one that is highlighted here, this is the attached Vibrio that was isolated. And in each band represents a species. So the blue marker here is the attached Vibrio. This is the one that was isolated using flow cytometry. And the red is the one that is the free-living Vibrio. And sequencing the band here, this was isolated. I determined this was Vibrio cholerae. So we can see that during a natural bloom of heterosigma, Vibrio cholerae is attaching to heterosigma. So the next step, usually I was hoping for another bloom to occur. That never happens whenever you want it to happen. So I had to go out and collect water and go and just gross filter it through a 20 micron filter. I then took that water and spiked cultures of heterosigma into it, and once again, or incubated for 24 hours, once again isolated using flow cytometry and analyzed that attached Vibrio community using TTGE. So once again, on the left here in the blue is our attached Vibrio community, and on the right is the free living. And isolating this and sequencing that particular band tells us this is Vibrio cholerae. So this is just demonstrating that between natural samples and using water from the Delaware Inland Basin, non-bloom conditions, and spiking heterosigma into it, we're seeing this association between Vibrio cholerae and heterosigma. Now I also carried out, sorry the images are not very clear here, other experiments using a transformed Vibrio cholerae with the GFP or green fluorescent protein plasmid transformed. And so we can see here at A and B, this is looking at all the fluorescence of the chlorophyll in red of heterosigma, and the green, this is your Vibrio cholerae attaching to those heterosigma cells. And C, the far right image, this is a DAPI stain, so this is a general nuclear stain. You can actually see the bright white spot in the center is the nucleus of heterosigma, and And all the little fine dots near the very bottom, those are all the Vibrio cholerae cells. And what is interesting to note is that it seems that all of these cells are actually attaching near the flagella, or aggregating around the flagellar pore of heterosigma. So now that I've seen this association and this attachment of Vibrio cholerae to heterosigma, what is Vibrio cholerae? What effects is the presence of Vibrio cholerae having on heterosigma itself? In order to do that, I carried out next generation sequencing, or RNA-seq. And to examine this, I looked at controls of heterosigma, cultured heterosigma by itself, and I spiked Vibrio cholerae into cultures of heterosigma. I incubated those for 24 hours, filtered them out, extracted the RNA, sent it up to DBI for sequencing. I assembled it using Trinity. Now, however, I should state that currently there is no sequenced genome for heterosigma. So that means that the transcriptome that I produced actually had to be assembled by using the software, using that as a reference transcriptome. And using that, I looked at differential expression using cufflinks and validated that differential expression using qPCR. Now, as I said, since this was a de novo assembly of a reference transcriptome, there's a lot of tears and frustration going into trying to assemble this. But looking at this, for the total number of reads, I had approximately 210 million reads in total. After QCing and cleaning it up, I came out with approximately 12,600, not approximately, but 12,698 contigs, or the number of sequences for this particular, for heterosigma. Now, comparing this number to other related species, this number falls within range of other related species. So this gives me a little bit of confidence that I'm close, or it gives me confidence what I did was actually very good. Now, looking at this, you can see other statistics on there, but the other important one here is this N50. This N50 number gives us an idea of the quality of our assembled transcriptome, and this is actually a very good number to have here at 1315. So since there is no sequenced genome for heterosigma, the next step was to annotate that reference transcriptome that I assembled. So to do that, I went through multiple steps, multiple programs to do this. One of them was using Trinotate, which is part of the Trinity package. Another one was using BlastX against the Marine Microbial Eukaryotic Transcriptome Sequencing Project, which was a very large project where they sequenced around 650 different microbial eukaryotic organisms. I also used MapMen, and this eukaryotic orthologous groups are called. So BlastX search against the unit product database and came up with about 37% of my contigs actually being identified. So that leaves about 67% or 63% of my contigs not currently identified. Now, looking at those BlastX results against unit product database, and how much of those sequences actually mapped to the full length of the sequences within the database is what we're seeing here on the graph here. So we're looking here at the percentage of full length from 100 down to 50, and the number of contigs there on the y-axis. So we see here, or you see on this graph here, there's about 2,800 contigs mapped in greater than 50% of coverage of the unit product database. So that gives me good confidence that about 3,000 or 2,800 of those contigs have a pretty good annotation to them. Now, looking at the COG analysis, this just gives us ideas of what the genes may be part of. We see that the COG analysis is divided into three sections. We have in green the cellular processing and signaling, as you can see by the pie chart here, this is looking at COG analysis of the entire transcriptome. This is taking up, you can see here, that this processing and signaling is about 50% of those contigs. The rest of the 50% is divided up between the blue, information storage and processing, and orange, metabolism. And you can see that the three major sections of the annotation here is in this single transduction, post-translational modification, and intercellular trafficking. So that's just very brief descriptions of my annotations that I'm going to go into. We're now just still delving into what is occurring with the transcriptome here. To examine differential expression, I used, as I mentioned earlier, I examined this using cufflinks, and I saw that there was 1,560 differentially expressed genes using a cutoff value, a Q cutoff value of 0.01. And the Q cutoff is slightly different than the P value. The Q is a modified P for false discovery rate. And of those 1,560, 1,001 contigs were upregulated, and the other 559 were downregulated. And this is just showing you a graph here of the up and downregulated. The blue are the significantly expressed, the red are non-significant. To validate the differential expression, I designed primers of a few sequences. We have fucoxanthin chlorophyll binding proteins, the glutathione peroxidase, glutaredoxin, this plasma membrane ion permease, and peroxidase. Using qPCR, I looked at the chain log differential expression of these different genes. And you can see here with this graph is there is a really good general agreement between cufflinks and qPCR. So this gave me really good hope that my differential expression analysis was actually, I was on the right track with that since they agreed very well. Obviously, if there had been somewhere up, qPCR saw upregulation, cufflinks saw downregulation, things would have been a little bit different. My next step was to look at a fairly new analysis called MapMan. The MapMan is an approach that is specifically designed for, or orientated towards, plants and algae. Now, the MapMan approach is very similar to using the Go approach, and that is takes the sequences and bins them into as few bins as possible that have different annotations to them. Right now, there are about 35 different categories for MapMan, and of them, dividing that even further, there's about 1,900 different bins within those categories. So here, I'm showing here the significantly expressed genes that were mapped into the MapMan here. Once again, you can see the majority of that is mapped into what they call the protein bin, and this includes things like modification, turnover, etc. Then we have transport, RNA, and etc. I'm not going to read into it. Now here, using MapMan, they also produce a really nice program that allows you to take those significantly expressed genes and then look at it in pathways throughout. And this particular one is a little bit more centered towards plant cells. But this here is looking at just a general overview of what is happening within the cell here. So the bars, the histograms that you see in green, these are significantly upregulated. Those in red are significantly downregulated. Taking this analysis a little bit further, I actually took this analysis and did some gene enrichment analysis with this. And so this is just looking at where are we seeing a lot of these, what bins are a lot of these genes being found in. And once again, this is showing here in the protein synthesis, the modification and degradation here I've outlined in red. We also see, once again, this vascular transport is another highly upregulated or enriched section, as well as the RNA processing, synthesis, and DNA synthesis. So, in conclusion, there is a very strong correlation between Vibrio abundance and abundance of rhapidophytes, specifically heterosigma, in Delaware inland bays. And despite this increase in grazing pressure, the association with particles can increase growth and potential proliferation, providing Vibrio species, or some Vibrio species, with an advantage. And using the transcriptome data that I just showed you here, can provide data to evaluate the responses to the presence of Vibrio and other marine bacteria in the environment. So the question is, what can be done next? So far, my transcriptome was looking at just within the laboratory environment. So the next question is, what is heterosigma doing during bloom conditions? So we can take a look at transcriptional response of heterosigma in these bloom conditions. So what is in the cell doing during the progression of the bloom? And you can also take a look at, if we size fractionate it again, and further look at what is the associated bacteria. How is that community changing during the bloom overall? And so with that, I would like to acknowledge the inland bays. Without them, I would not be doing this research here. Of course, the Coin Lab picture shown here, even though Charles wasn't really part of the Coin Lab, he did spend an awful lot of time in our lab. Of course, the faculty and staff, I really like to say, I'm sure all you graduate students know, on Wednesdays, Maxine, make sure to say very nice things to her, because she's the one that feeds us every Wednesday. So please thank her as much as possible. But of course, Connie, Debbie, Ellen, Marcia, Janice, and Lisa, all of them, I could not do this without that. And of course, my friends and family and other graduate students that shared out today. And with that, I'll take any questions that you may have.
Okay, do we have any questions for Chris?
That's never a concern. Joanna, sorry, your hand first.
So you said that the microzoa function crazy can be deterred by association with particles sometimes. Do I understand?
No, so the microzoa, it's not a deterrent, it's more of that it's providing a refuge. So that image that I showed you in the very beginning of the phycosphere, they're releasing those nutrients out into the marine environment. So it enriches and increases their abundance. So the idea is they're attaching to these particles that they're proliferating much quicker than they're actually being grazed.
So that's why you see this.
Yeah. So
Yeah, it's kind of, perhaps you don't have an answer if you don't know oysters, but how well does an oyster filter the water? Would the oyster tend to pass through any Vibrio that's in this tiny fraction and not really be able to take it up? And so would most of the, risk of a shellfish-borne illness, could you guess that it would be related to eating a Vibrio attached to algae?
well, I'm not very familiar with oysters, but I do know that they do sequester, when they do filter out, they do sequester a lot of the Vibrio within their stomach contents.
Right.
So I'm assuming, yes, if we're seeing this correlation between, as blooms increase, and they are just starting to reintroduce oysters for oyster farming, aquaculture in the bays, there could be a chance that there may be more increases of Vibrio from eating oysters and things like that.
Is this, do you think this is a passive interaction between Vibrio and other algae, or do Vibrio have the capacity to harm them?
That's actually a really good question. And that's actually something that would be really interesting to explore, because right now all my research focuses in on the algal aspect of it and nothing on the Vibrio aspect of it. And there could be some kind of antagonistic. There have been publications of unknown Vibrio species. They're just Vibrio, not unidentified at the species level, that have been shown to as an algicide. If I'm not mistaken, there was one found off of South Carolina that was actually shown to be an algicide against heterosigma, as well as chatnone, I think maybe fiber capsid, but I know heterosigma for sure. So in certain parts, it may actually be an antagonistic relationship.
So along that lines, what do you make of this Vibrio's associated anthoflagellar foreign? Like, do they give you any suggestions that it's maybe not just an accidental?
Yes,
Or
I
does it
mean,
make
I don't, well, that's just I don't know. I mean, that's one of the things that we've seen it. We need to figure out why that's occurring, why it's happening, what is it about the flagellant report that why are they aggregating there? And I'm not sure.
Has there been an indication if that's where the exudates are most concentrated?
Well, one of the things about the raphidifies is they lack that cell wall.
Mm-hmm.
So, well, actually, there could be an area of concentration of exudates there, but I don't think anybody's ever looked into it, or at least I haven't ever come across it.
Do we know how the Vibrio bacteria actually physically attach to their algal host, if you will? I mean, if you washed the algal cells over and over again, would you see loss of Vibrio, or are they really stuck on there?
There's potential. So, looking at it under the microscope, I have seen where with live Vibrio cells in the heterosigma, they'll come in, attach very briefly for about 10 to 15 seconds, and swim right off, and just do that multiple times. Fortunately, it was always when I went to go grab, they had captured. Of course, that never happens to get a good image of it. Now, some of them actually, heterosigma can produce a EPS, so that may be an area of aggregation occurring that they're producing, getting called this EPS. There's actually been some research shown that sinking rates of heterosigma actually increase their toxicity, and there's actually been shown that this EPS, there's actually been extracellular material from a heterosigma that also may be linked to their toxicity as well. So there's a few things that, but the Vibrio also do form biofilms within their environment. However, there is this kind of cutoff point with the biofilm that you reach a certain point that you can't detach from it. So there's a trade-off between all of this. But my guess would be it's probably more of since we're in the marine environment, it's going to be more of a kind of fluid association.
Any questions from Newark? Anybody else? Okay, if there aren't any more questions, we're going to follow the standard format. Thank you all for coming, and we'll take a brief recess and then reconvene with the committee members. I believe it's happy hour in about 15 minutes downstairs. So
We can already start that.
Well, it's already
we can get it started now. Yeah. And then, all right, so thank you all for coming.
How are you doing? I thought you were going to be Skyping in.
I considered it, but as I was talking about, I think that for a PhD, I'd like
Yeah, it's nice
to
building.
be there.
Yeah.
Present if it was master's, I probably would have been
Yeah.
But also, happy I had some more
Yeah.
about some of our funded projects.
It's a good excuse to get together.
Yeah. But no, I think for PhD,
Yeah.
if possible, should try to make it. Besides, it's always good to come up here.
Did you drive up?